Welcome to another lecture by MedicoMedics, Microbiology and Infectious Diseases, Chapter 5, Mycology, the study of fungi. So in this lecture, we will do an introduction to fungi, discuss structure, classification, some infections, diagnosing fungal infections. We will mention antifungal agents, and then continue down to end with a summary. So fungi are eukaryotic organisms with cell walls made of chitin. And chitin is a natural polysaccharide made up of acetylglucosamine, essentially a derivative of glucose molecules linked together. And it's primarily known for its role as a structural component in various organisms. And in fungi, in the cell walls of the fungi, it provides with structural integrity. Now let's mention some types of fungi. So we have yeasts, right? These are single-celled fungi. An example is Candida. We have molds, so multicellular fungi with filamentous structures like Aspergillus or Aspergillus. Then we have dimorphic fungi. So this switches between yeast and mold forms, like histoplasma. Now, why is this important? Because these are decomposers in ecosystems, and they are producers of medicines like penicillin. So all in all, fungi are like recyclers of nature. They're breaking down dead things, and they come in different forms, like single cells, yeast, or filaments, molds, and some of them are used to make medicine, others may cause infections. Now let's look at the structure of fungi. So key features, we mentioned a cell wall made of chitin. It's not made of cellulose like in plants. And we have hyphae, so filamentous structures that form mycelium, the fungal body. Another important feature are spores. So these are reproductive units for spreading and growth. And we have types of hyphae. So we have septate, divided by walls, and choanocytic, where we have no internal walls. Now, how do we classify fungi? Well, we have these major groups: Zygomycota, Mycota, bread molds like Rhizopus, Ascomycota, sac fungi like Candida or Aspergillus, Basidiomycota, club fungi like mushrooms, and let's say that's Chytridiomycota, aquatic fungi with flagellated spores. And the classification is based on reproduction. So either we have sexual reproduction, so fusion of spores, or asexual reproduction, so spores produced by mitosis. So what's important to remember is that fungi are grouped based on how they reproduce. Now, fungal infections or mycoses. These can be subdivided into different categories. So we have superficial mycoses. These affect hair, skin, and nails, like athlete's foot. We have subcutaneous mycoses affecting deeper skin layers or tissues. Systemic mycoses, so affecting internal organs like histoplasmosis. And opportunistic mycoses affecting immunocompromised people like Candida albicans. Transmission, direct contact, or inhalation of spores, or environmental exposure. So fungal infections range from annoying skin problems to serious internal infections, especially in people with weak immune systems. And these infections often spread, right, through contact or breathing in of these spores. Now, how do we diagnose fungal infections? Well, the methods we use include microscopy, so we do staining and viewing under a microscope. Culture test, growing fungi on a special media. We do molecular techniques like PCR to detect fungal DNA. Some examples like the co-test, dissolving human cells to highlight the fungal cells, the India ink stain detecting Cryptococcus in cerebrospinal fluid. Now, let's talk about some antifungal agents. And antifungal drugs target the unique parts of the fungi, like their walls or membranes. But treating fungal infections can be tricky because their cells are a lot like ours. So treatment needs to be very careful and specific. But what kind of types do we have? Well, we have azoles. These inhibit cell membrane synthesis, like fluconazole. We have polyenes. These bind to fungal membranes, causing cell death. We have something called echinocandins. These inhibit fungal cell wall synthesis. And if you remember, if we talk about challenges, right? And it says fungal infections are harder to treat than bacterial ones due to similar cell structures to human. Do you remember some specific things in bacteria that makes it, in a way, easier to target and to differentiate from human cells? Well, if you remember in our lecture, we talked about the ribosomes that are involved in the creation or production of proteins, right? And that we identified in bacteria, we have different subunits compared to eukaryotic cells. So these prokaryotic cells had the smaller subunit of 30S. If you remember, S was Swedberg unit, and 50S, compared to humans that had 40S and 60S. That's not an S. And that eukaryotic cells are put into 80S in total, and bacterial is 70S. And this differentiation helps us in developing drugs to specifically target these different things without actually targeting human ribosomes or preventing human synthesis of proteins, etc. Now, in fungi, this is a bit tricky because we don't have this same differentiation, so it is indeed harder to treat. Fungi and the environment. Ecological roles. So fungi are nature's sort of cleanup crew. They break down dead things and help plants to grow. But they're also sort of troublemakers in agriculture, specifically causing plant diseases that can actually ruin crops. But some roles include decomposers, indeed recycles nutrients by breaking down dead matter. Symbiosis, so it partners with plants for better nutrient absorption. And of course, pathogens, so it causes diseases in plants. And now let's turn to fungi in medicine. So applications. We use it in antibiotics. An antibiotic known to most people is penicillin, and it is derived from Penicillium, a fungi. We have immunosuppressants like cyclosporine, used for organ transplants, used in vaccines like Candida albicans, used for research in antifungal vaccines. Then we have fungal toxins, aflatoxins. So these are harmful substances produced by Aspergillus. So all in all, fungi are both sort of heroes, but also villains when we're talking about medicine. They give us life-saving drugs, like antibiotics, but also produce harmful toxins that can contaminate, for example, our food, etc. A bit of a yin and yang of nature. Some industrial applications of fungi. So it's used in fermentation, yeasts in bread, beer, and wine production. Bioremediation or remediation, breaking down pollutants in the environment. Enzymes, so industrial production of cellulases and proteases. What about fungi and biotech? Some applications include producing biofuels from plant waste, involvement in genetic engineering. Let's say that as a model organism, the production of recombinant proteins like insulin. So in summary then, fungi are diverse and important in ecosystems, in medicine, and in industry. While some fungi are beneficial, like penicillin, others can cause serious infections or even crop damage. Understanding fungi helps us fight diseases and use them in innovative ways. So fungi are everywhere, essential for life, but can also cause problems. By studying them, we can harness their power for good while protecting ourselves from harm. And that's the end of chapter 5. Continue now to chapter 6.